Welcome to this class on neuroscience of human movement. In today's class, we will be discussing basal ganglia, in particular the outputs of the basal ganglia. So, in this class, we will discuss major output structures. We have already mentioned what these are. This is the globus pallidus internal segment and the substantia nigra pars reticulata, and the outputs from the thalamus to the cortex, which are basically the ones that are modulated by the basal ganglia output, right, and outputs to the brainstem motor nuclei, and indirect outputs to the spinal cord through brainstem areas. In other words, brainstem in this case acts as the upper motor neurons, right, or motor areas in the brainstem act as the upper motor area for these cases. Something that we discussed yesterday, but we missed in yesterday's class, are these corticostriatal pathway and inputs. Importantly, not only the corticostriatal pathway, so there are exclusive projections from the cortex to the striatum, right. Other than these, cortico-cortical projections, corticospinal projections and corticothalamic projections are those that go from one area of the cortex to another area of the cortex, or from the cortex to the spinal cord, or from the cortex to the thalamus. These axons also send their branches or collaterals to medium spiny neurons. So, they terminate on medium spiny neurons, thus exciting them. Let us remember that medium spiny neurons are GABAergic and fundamentally inhibitory. If they are excited, they are going to inhibit their target neurons, but they are usually quiet. Why are they usually quiet? Why are they quiescent? Because of the inward rectifier potassium conductances, that is potassium K plus, inward rectifier potassium conductances that keep the membrane potential near resting membrane potential, but these conductances these rectifiers close at depolarization. So, because of this unusual potassium conductance, it turns out that the medium spiny neurons are usually quiet, and they need input from multiple cortical or nigral neurons to activate, right. And we also saw what are the various kinds of inputs it receives, that the inputs received from the cortical and the nigral neurons are actually at the distal end of the dendrites, whereas closer to the soma and cell body there are actually inputs coming from local interneurons and from thalamic neurons. We have seen this. So, in that sense, the local interneurons modulate the output of the medium spiny neurons in a great way when compared with the cortical neurons. So, multiple cortical neurons must send their input to the medium spiny neurons for them to be excited. Spatial and temporal summation must happen, and then action potential will be generated in the medium spiny neurons. This is the case that we discussed yesterday. But regarding the cortical connections itself, connections from one cortical neuron to an individual medium spiny neuron is relatively small. So, basically each cortical neuron sends one input to one medium spiny neuron, or approximately that much, not too many connections to each medium spiny neuron. However, each cortical axon contacts many medium spiny neurons. So, what happens is that one cortical neuron sends multiple collaterals to multiple medium spiny neurons, and one medium spiny neuron receives inputs from multiple cortical axons. So, what is there in this situation is that there is a great divergence, of the cortical information onto many medium spiny neurons, and we saw yesterday that medium spiny neurons converge onto the pallidal neurons, whereas from the cortex there is in general a great divergence into the medium spiny neurons. This enables integration of information from multiple cortical neurons by the medium spiny neurons, right. And activity of the medium spiny neurons has been studied, and people have asked the question, what causes the medium spiny neurons to fire, what activates them? In general, it has been found that the destination of the movement, suppose I have this object, I have to reach this object, where this object is determines which striatal neuron is going to be active, not which limb is reaching or what movement is going to be done. So, this is, to use a strong word, this is encoded in the space of the target location, not in the space of limb dynamics. So, that means essentially what the basal ganglia, what the striatum encodes is a decision to move. First, whether or not to make a movement. If the decision to make a movement is made, what movement must be made? In other words, to which target must we move? A decision is what is being encoded by the striatal neurons. With this, we finished the earlier discussion on inputs. Now, we will move on to the major output structures. We have discussed this earlier. We said the major output structures of the basal ganglia are the globus pallidus internal segment and the substantia nigra pars reticulata. Essentially, the substantia nigra pars reticulata and globus pallidus internal are the same output structures separated by the internal capsule. Note that the substantia nigra pars reticulata, unlike the compacta contains compact packaging of cells which are composed of the many of these are dopaminergic in the substantia nigra pars compacta. But in the reticulata, the axons passing through the pars reticulata give the reticulata an appearance of net like that, right. So, because of this reason, this region is called as reticulata, substantia nigra pars reticulata. Both of these are the major output structures of the basal ganglia, right, and these are separated by the internal capsule. In other words, substantia nigra pars reticulata can be called as the downward displaced GPI, downward displaced globus pallidus internal, right. And we already said that the axons from medium spiny neurons from the striatum converge onto the pallidal neurons, right. On an average 100 medium spiny neurons converge onto one pallidal cell. What is also to be noted is that there is a broad spectrum of outputs to the pallidal neurons. So, what happens is that medium spiny neurons send outputs to multiple pallidal neurons, right. So, they make synapses with multiple pallidal neurons, but one pallidal neuron gets inputs from hundreds of medium spiny neurons. So, the situation is as follows. The situation is that there is a broad inhibition that is provided by the striatum to the pallidal neurons. When the striatum itself is excited, that excitation converts into a relatively sharp inhibition. So, there is in general a broad activation of the pallidal neurons or a broad connections that are made by the spiny neurons onto the pallidal neurons, but some of these are sharp. In other words, medium spiny neurons project to several neurons at the same time, but to some of the neurons they make very specific strong and sharp connections. Because of this reason, when a particular medium spiny neuron is active, a particular pallidal neuron is going to be inhibited from its activity. This is the way in which this functions. Is somewhat similar or at least comparable to our discussion in cerebellum about the Purkinje cells, right. Something that we should remember, that there is sharpening of focus in the Purkinje cell by the parallel fiber inputs, right, something that we discussed some time ago in the previous classes. So, let us remember, so there is divergence from the cortex into the striatum, and then there is convergence from the striatum into the pallidal neurons, and the pallidal neurons are the major output neurons, right. So, they output to the thalamus, or the substantia nigra pars reticulata outputs to the superior colliculus, right. The GPI neurons output to the thalamus, and they thus inhibit the thalamocortical neurons from being active, something that we will discuss in future slides, right. So, the thalamocortical outputs here, thalamus or the neurons in the VA-VL complex of the thalamus act as relay. They take the output of the GPA, they take the output of the basal ganglia and inform the cortex of the basal ganglia's modulation, right. So, from the VAVL complex, from the ventro-anterior and ventrolateral complex of the thalamus, there are projections to the cortex. When you say cortex, we will have to ask ourselves which cortex this is, right. So, regardless of where the input is coming from to the basal ganglia. So, the basal ganglia may receive input from practically anywhere, right, in the cortex, except the primary auditory and primary visual cortexes. Practically from everywhere else basal ganglia is receiving input, but its outputs are always to the precentral areas, to the frontal cortices, something to remember, either to the motor areas or to the executive and association areas of the frontal cortex or the prefrontal cortex. This is where the basal ganglia's output goes to, not to other areas in the cortex. For example, not to the primary somatosensory cortex, for example, right. So, also what happens is from the striatum, there are neurons that inhibit substantia nigra pars reticulata, and they directly project in one case to the superior colliculus. Some of this substantia nigra pars reticulata also projects to thalamus, and from thalamus to the frontal eye fields. Once again, the output is to the frontal area of the cortex, FEF in the frontal cortex, right, something to remember. So, let us look at the situation one more time. So, basically there is output from the GPI, which is here. So, this is putamen, sorry, this is GPE, and this is GPI, right. From there the output goes to the VAVL neurons here, and from there back to the cortex, right. So, that is what happens, and if it is from the substantia nigra pars reticulata, the output goes to the superior colliculus, right, for controlling eye movements. So, the output to the brain stem, other than the cortical areas outputs, there are also outputs to the brain stem, right. This is useful in the control of eye movements, head movements, balance and gait. So, from the striatum there is output to the substantia nigra pars reticulata, which is the output structure. From there the output goes directly to the upper motor neurons of the superior colliculus without a relay in the thalamus, right. In the case of substantia nigra pars reticulata, there are many projections that go directly to the brainstem, directly to the superior colliculus without a relay in the thalamus. Whereas, if there is an output that is going to the frontal cortex from the substantia nigra pars reticulata, for example, in the case of frontal eye fields, then what happens from the substantia nigra pars reticulata, there is an output that goes to the thalamus, and through the thalamus relay it goes to the frontal eye field, as we discussed. So, something to remember, that if it is projecting to the brainstem, there is no relay in the thalamus. If it is projecting to the cortex, there is a relay in the thalamus, right. So, from the striatum, substantia nigra pars reticulata or GPI project to the pedunculopontine nucleus to basically control balance and gait. In this case, there is no relay in the thalamus that is involved, crucial to note this point, right. So, essentially brainstem may integrate outputs that come from the basal ganglia and also from the cerebellum. Importantly, brainstem receives inputs from the cerebellum also. So, the control of balance and gait is managed not just by the cortex, by also these subcortical structures, basically cerebellum, basically basal ganglia, right. And also note, pedunculopontine nucleus sends inputs to the basal ganglia via the thalamus, something that is not generally mentioned. Pedunculopontine nucleus is also part of a feedback loop that sends inputs to the basal ganglia via the thalamus, right. So, essentially the efferent neurons, the output neurons of the globus pallidus internal and the substantia nigra pars reticulata are GABAergic, and so that means their main output or the main output of basal ganglia is in general inhibitory. So, and this means that they provide tonic inhibition to the thalamus or the thalamocortical neurons and the superior colliculus. So, essentially what they are doing is they are tonically inhibiting, preventing unwanted movements. An immediate hypothesis that we can make is that if this function of the output neurons is somehow selectively compromised, then what will happen? They are tonically inhibiting, preventing unwanted movements. Suppose this function is compromised, then what will happen? Well, immediately unwanted movements will start appearing, right, like in the case of chorea. This is essentially what happens in Huntington's chorea, something that we will discuss in future classes, right. So, selective degeneration of the indirect pathway leads to Huntington's chorea, something that we will discuss. Essentially unwanted movements are prevented by this tonic inhibition, and what ensures that the wanted movements are performed, the intentional movements are performed. What ensures that? That is ensured by transient selective disinhibition that comes from the striatum. So, striatum essentially inhibits for a brief period of time selectively some movements alone. Now, that opens the gate for that movement to appear, all the other movements are gated. This is the interesting and the dominant hypothesis in basal ganglia function, right. By the way, that also means that malfunction of this circuitry means chorea, right, something that we discussed. A lot of this evidence to support the presented ideas come from eye movement studies. I will draw the picture for this and we will discuss this in a minute. So, here I have drawn this situation, a very simplified situation of what happens in the basal ganglia, very cartoon type of diagram drawn with my own hand, but it is going to explain the principle, right. So, essentially what is happening is that the medium spiny neuron in the striatum, which I am going to call as neuron 1 N1, is receiving inputs from multiple cortical neurons which are shown in green here. The neurons in green are the cortical neurons, and the neuron in yellow here is the substantia nigra pars compacta neuron or the dopaminergic neuron. So, this medium spiny neuron is receiving input from several cortical neurons and some nigral neurons, and this is making synapses with pallidal neurons, neurons in the globus pallidus, right, which I am calling here as N2, right. N2 or the neuron 2 is the pallidal neuron, which is inhibiting, which is tonically inhibiting the neuron in the ventro-anterior or ventrolateral thalamus, and this neuron is called as the neuron 3 or N3, and that is exciting the cortical neuron which I am calling as neuron N4. Let us consider this is a neuron in the motor cortex, right. N4 is a neuron in the motor cortex. For the purpose of this discussion, we will consider N4 to be a neuron in the motor cortex, right. So, essentially suppose neuron 1 is at rest, its activity is like this, right. What will be the inhibition provided by neuron 1 to neuron 2? Nothing, because neuron 1 is quiescent, it is normally quiescent. Let us remember that medium spiny neurons are normally quiet because of unusual potassium conductance, is something that we saw, right. N1 is not inhibiting N2, and because of this N2 is tonically active and tonically inhibiting the thalamocortical neuron or the neuron in the thalamus, right. So, essentially the activity of N2 will be very high, like this, continuously it will be like this. For that time period that is recorded, this will be flat, N1 will be flat, N2 will be continuously active. If N2 is continuously inhibiting N3, note the output of the pallidum is essentially inhibitory, right. It is continuously sending GABA to N3, right. Because of that reason N3 is not active, N3 is silent, and because of that reason N4 is also silent. Why? That means if the striatal neuron is quiet, that means that the motor cortical neuron is also quiet, preventing unwanted movements. Suppose I want to produce a movement, what happens is that N1 neuron becomes transiently active, right. So, what happens is that this is flat for some time, but is active for a brief amount of time, say that. Then what happens in the pallidal neuron? Here is what happens. So, essentially for that time period alone there is silence in the pallidal neuron. So, during the time for which the striatal neuron is active, the pallidal neuron is silent. When the pallidal neuron is silent, what happens in the thalamus, in the neuron in the relay at the thalamus? Usually it is silent. During the time during which the pallidal neuron is silent, that is excited. For that time alone it is excited, and for that time alone the neuron 4 is excited, thus performing the intentional or movements that are needed as opposed to producing unwanted movements, right. So, this is essentially what happens. So, the cardiac nucleus neuron is usually silent, and when it is active for a brief amount of time, it causes, so this is caudate, this is substantia nigra pars reticulata. Substantia nigra pars reticulata neuron is continuously active, and when the cardiac nucleus neuron is active, it is silent. When the cardiac nucleus is active, the substantia nigra pars reticulata is silent, and then it is active again. Is usually silent. During the time when the substantia nigra pars reticulata neuron is silent, the superior colliculus neuron is active, and at other times it is silent. This activity causes eye movements, right. Essentially this is how a specific eye movement is made, right. However, note in this case there is no relay that is in the thalamus. So, this is simpler, is an easier model to study. Lot of data from animal models, especially monkeys, have been performed by Okihide Hikosaka and Robert Woods. Hikosaka and Woods, this is data that I am trying to reproduce from the work of Hikosaka and Woods, right. So, essentially this is what happens. Essentially the transient activation of the striatal neurons by the cortex transiently inhibits the pallidal neurons, which are usually tonically inhibiting the thalamus neurons, thus releasing the thalamus neurons from inhibition for a brief period of time or causing disinhibition. So, basal ganglia or striatum is essentially disinhibiting the thalamocortical neurons for a transient brief amount of time, thus producing intentional or volitional movements. So, in summary, what we have seen are the basal ganglia's major output structures, which are globus pallidus internal and substantia nigra pars reticulata, and the outputs to the cortex are gated by the thalamus or through relays in the thalamus, and outputs to the brainstem and superior colliculus are usually not gated, and substantia nigra pars reticulata, when it sends information to the frontal areas in the cortex, it sends it via the thalamus, right. Output of basal ganglia is fundamentally output of the basal ganglia is fundamentally inhibitory or GABAergic, and essentially whenever striatum initiates activity, it initiates movement by this inhibition. With this we come to the end of this lecture. Thank you very much for your attention.